Welcome to another deep dive, listeners. Buckle up, because today we're tackling something pretty complex, cannabinoid receptor signal transduction.
Yeah, we're going to try to break down how these receptors work, how they make things happen inside your cells, when cannabinoids enter the picture.
And we're not just talking about THC from cannabis, right?
Mm-hmm.
We're talking about your body's own endocannabinoids, too.
Exactly.
To guide us, we've got some heavy hitters in the research world. We're talking Dr. Ethan Russo, Dr. Raphael Mechoulam. These folks are like the trailblazers of cannabinoid science.
Real pioneers, yeah.
Okay, so to start, can we just visualize these cannabinoid receptors? Where are they? What do they look like?
Well, imagine tiny little antennas on the surface of your cells. That's them, CB1 and CB2, constantly scanning, waiting.
And they're tuned to a specific frequency, right? Cannabinoids.
Exactly. They're part of a family, a group of proteins called G-protein-coupled receptors, or GPCRs. Think of them like gatekeepers at the cell membrane.
Controlling what gets in and out.
Right. So when a cannabinoid, let's say THC, binds to the receptor, it's like a key fitting into a lock. It changes the shape of the receptor.
So there's movement. It's not just a static thing.
Oh, yeah, definitely. And that movement, that shift, it kicks off a chain reaction. It activates these molecules called G proteins.
G proteins, those are the messengers inside the cell.
So that's them, yeah, like a relay team.
Sure.
The signal gets passed from the receptor to the G protein, and then
Off it goes to different parts of the cell.
Right. And depending on which G protein is involved, the signal takes a different route, a different pathway.
So multiple paths, not just one straight line.
Exactly. It's like a network of roads. One major pathway uses a G protein that puts the brakes on a molecule called cyclic AMP.
CMP. Now, didn't I read somewhere that CANMP is involved in a ton of stuff inside the cell?
Oh, absolutely. By changing CANMP levels, cannabinoids can mess with a whole bunch of cellular activities. Mood, appetite, pain, even memory, you name it.
Wow, that's a lot of influence. Okay, so that's one pathway. What about the other route the signal can take?
The other main pathway, it uses a different G protein. This one flips the switch on an enzyme called phospholipase C, or PLC.
PLC, okay. And what does this enzyme do?
Think of it like a chef chopping up a big molecule into smaller pieces.
A molecular butcher, kind of?
Yeah, you could say that. The pieces it creates are called DAG and IP3. These little guys then go on to activate other pathways, things like calcium signaling.
Calcium signaling, that rings a bell. Isn't that involved in muscle contractions and nerve impulses?
You got it. And by affecting calcium, cannabinoids can really change how cells behave.
Man, this is getting complicated, but I'm starting to see how all these different pathways can lead to such a wide range of effects throughout the body.
Yeah, it's a complex system for sure. But wait, there's more.
Oh, no, more.
Yeah, so we talked about G proteins, but there's another key player in this whole signaling cascade, beta-arrestin.
Beta-arrestin. Okay, I remember reading about that. It acts like a brake, slowing down the G protein signal, right?
That's one way to think about it, yeah. But it can also do its own thing. It can activate completely different pathways.
Wait, so it's not just a brake pedal. It can steer the car in a whole new direction.
Exactly. It's like having a key that can unlock multiple doors in the cell.
Wow, talk about multitasking.
Yeah, beta-arrestin is fascinating. It adds a whole other layer of complexity to how cannabinoids work, and it's got researchers really excited.
I can see why. So much potential there. Okay, we've covered a lot of ground here just on the basics, but I'm really starting to see how intricate this whole signaling process is.
And we're just getting started.
Oh boy, I'm ready for more.
Let's dive deeper then.
Okay, I'm ready for more. What else makes this beta-arrestin so interesting?
Well, it's like, you know how we were talking about all these different pathways, right? It adds even more possibilities. Like, it could be the key to, unlocking new therapies, new ways to use the endocannabinoid system to treat diseases.
Like a secret passage, leading to a whole new wing of the house.
Yeah, exactly. And if we can understand how all these pathways work, how beta-arrestin fits in, then, we can really start to design better treatments, ones that are more targeted and effective.
So it's not just about turning a pathway on or off. It's about understanding how all these different players interact with each other.
Exactly. It's like an orchestra. You need all the instruments playing in harmony to create beautiful music, right?
Right. And if one instrument is out of tune, the whole piece can fall apart.
Exactly. So we need to understand each instrument, how they work together. So let's look at some of the specific molecules involved, like kinases, for example.
Kinases. Oh, yeah, I remember those. They're like tiny little switches that can turn other proteins on or off.
You got it. They add these things called phosphate groups to proteins, and that can change how those proteins behave, like flipping a switch.
Molecular light switch.
Yeah, exactly. And there are a bunch of these kinases that are super important when it comes to cannabinoid signaling.
Like which ones? Give me some names.
Well, you got your MAPK, your ERK, JNK, P38, just to name a few.
Okay, those are ringing a bell, but I'm a little fuzzy on the details. Remind me what they do.
Sure. These guys, they're involved in all sorts of important cellular processes, like how cells grow, how they differentiate, even how they die. Big stuff,
Wow, so they're not just bit players, huh?
Oh, no, they're calling the shots. And research has shown that cannabinoids can actually influence how these kinases work.
So cannabinoids are kind of like the conductors of the orchestra, telling these kinases when to play and how loud.
Yeah, that's a good analogy. It's all about fine-tuning the whole system. But the thing is, with so many different kinases and pathways involved, it can be really tough to figure out how it all works together.
It's like trying to solve a giant puzzle.
Exactly. And then you throw in something like bias signaling, and it gets even more complicated.
Bias signaling, what was that again?
Well, remember how we were saying that a cannabinoid can bind to a receptor and activate different pathways? Well, bias signaling means that some cannabinoids, they might prefer to activate one pathway over another.
So it's not just about hitting the receptor, it's about hitting it in just the right way to get the effect you want.
You got it. And this is huge because it means we could potentially develop drugs that are way more specific, that target only the pathways we want and avoid the ones we don't.
So less chance of side effects.
Exactly. That's the hope anyway. And researchers are really digging into this, trying to find those special cannabinoids that can, hit the right notes and create the perfect harmony.
So it sounds like a real game changer for drug development.
It could be, yeah. But there's still a lot we don't know about biased signaling. Like, how do we even identify these special cannabinoids? How do we know which pathways to target? It's a big puzzle.
A puzzle with a lot of pieces.
Yeah, but it's a puzzle worth solving. Because if we can crack the code, we could unlock some amazing new treatments for all sorts of diseases.
Okay, so I think I'm starting to get this whole signaling thing, all the different players and pathways. But how does all this, microscopic stuff actually affect us, in the real world?
That's a great question. I mean, it's all well and good to talk about these molecules and pathways, but what does it all mean for, how we feel, how our bodies work?
Okay, so we've been talking about all this signaling, but, where's the action happening? Where are these cannabinoid receptors doing their thing?
Well, they're all over the body, but one place they're really concentrated is in the brain.
The brain, huh? So they must be playing a pretty big role up there.
Oh, yeah, huge role. Think about it. The brain is all about communication, right? Neurons sending signals back and forth.
And these CB1 receptors, they're right there on those neurons, eavesdropping on all the chatter.
Exactly. They can actually influence how those neurons talk to each other. You see, when a cannabinoid, like THC, binds to a CB1 receptor, it can change how much of a certain neurotransmitter gets released.
Neurotransmitters, those are the chemicals that carry messages between neurons, right?
Yep, that's them. So by messing with neurotransmitter release, cannabinoids can have all sorts of effects on mood, memory, perception, all that good stuff.
So it's like cannabinoids are tweaking the volume knobs on different brain circuits.
That's a great way to put it. It's all about fine-tuning the symphony of activity in the brain.
Wow, it's amazing to think that these tiny molecules can have such a big impact on how we think and feel.
It is, isn't it? But it's not just the brain. Remember CB2 receptors?
Yeah, those are mostly found on immune cells, right?
Right. So cannabinoids can also affect the immune system, how it fights off infections, how it responds to inflammation.
So the same system that can make you feel relaxed or giggly can also help your body fight off a cold.
Exactly. It's pretty amazing how interconnected everything is, and we're still just scratching the surface of what the endocannabinoid system can do.
It's like a whole secret world inside our bodies that we're only just beginning to understand.
Exactly. And the more we learn, the more potential we see for using cannabinoids to treat all sorts of diseases.
Okay, so there's a lot of excitement about the therapeutic potential, but I have to ask, are there any risks involved in messing with these pathways? Could there be any unintended consequences?
That's a great question, and it's something researchers are always thinking about. The endocannabinoid system is complex, and it's all about balance. If you tweak one pathway too much, you might throw off the whole system.
Like a domino effect.
Exactly. That's why it's so important to do careful research to understand all the different pieces and how they fit together before we start using cannabinoids as medicine.
It's like trying to fix a watch without knowing how all the gears work. You could end up breaking it.
Right. But thankfully, scientists are developing all sorts of new tools and techniques to study these pathways in detail.
Like what?
Well, one thing they're using is something called selective pharmacological tools. These are basically drugs that are designed to target very specific pathways.
So it's like having a set of tiny screwdrivers, each one designed to adjust a specific part of the watch.
Yeah, that's a good analogy. By using these tools, researchers can isolate and study the effects of different pathways.
So they can figure out which pathways are responsible for which effects.
Right. And then there's genetic manipulation. Scientists can create animals or cells that are missing certain genes, genes that are involved in the endocannabinoid system.
So they can see what happens when a specific part of the system is taken out.
Exactly. It's another way to pinpoint which pathways are important for which effects.
It sounds like there's a lot of exciting research going on.
Oh, yeah, definitely. And as we learn more, we'll be able to develop better and safer treatments that target the endocannabinoid system.
Well, I'm definitely going to be keeping an eye on this field. Thanks for taking us on this deep dive. It's been really fascinating.
You're welcome. And remember, we've only just scratched the surface here. There's so much more to explore and discover about the endocannabinoid system. So keep learning, keep asking questions, and keep those brains buzzing. We'll catch you on the next deep dive, folks.